Good afternoon. Welcome to this week's Wednesday webinar. We'll just give people a few minutes to join before we get started. Thank you for joining us today. We'll give people another minute or so to get logged in, and then we'll get started with this week's Wednesday webinar. Good afternoon, everyone, or good evening, or good morning, perhaps, depending on where you're joining us from. Thank you very much for joining us again this week for the weekly Wednesday webinar series. My name is Fiona Robinson, and I am the host of this series on behalf of the National Hemophilia Foundation. Today's webinar, we will be learning about gene editing. Please feel free to ask any questions you may have throughout the presentation by using the Q&A feature at the bottom of your screen. We have staff members monitoring these questions, which we will present to our speaker after the presentation. This webinar is being recorded and will be available to the community beginning on Friday, July 21st. Today, I'm joined by Professor Julian Gilmore. Professor Gilmore is the centre head at the UK National Amyloidosis Centre based at University College London. I'll turn things over to you now, Professor Gilmore, if you'd like to start.
Thank you very much, Fiona, and thank you very much for asking me to speak today. They always say, don't start a talk with an apology, but I will do. And I should start by explaining that I work at the UK National Amyloidosis Centre and I'm a so-called expert in amyloidosis, but I'm by no means an expert in gene editing technology, although I've of course been exposed to it in recent years. Be delighted to answer any questions at the end of the talk, as Fiona mentioned, but I'll hold my hand up now and let you know that there may be questions about recent advances in gene editing that, that I'm unable to answer. Anyway, it's a great pleasure for me to present to you the interim data from the first in-human trial of an in vivo CRISPR-Cas9 based gene editing therapy known as NTLA-2001. And that's designed to edit the TTR gene, the transthyretin gene, in patients with transthyretin amyloidosis. And this trial represents the first time any human has been given an infusional in vivo CRISPR-Cas9 based gene editing therapy. So just to introduce you to transthyretin amyloidosis, it may be either acquired or hereditary. Acquired transthyretin or ATTR amyloidosis is known as wild type, and it's a cardiomyopathy which is caused by amyloid deposits, which are composed of the protein transthyretin, which accumulate within the heart. It's an increasingly recognized cause of heart failure in the over 50s, and is almost certainly the most common type of amyloid, although the diagnosis continues to be missed. The panel on the right here shows the huge increase in the number of diagnoses in our national centre in recent years, with the red line representing patients with wild-type ATTR amyloidosis. And this has been the experience all over the world, and it's due to a combination of improved diagnostic imaging techniques and a greater disease awareness amongst cardiologists. In the absence of therapy, cardiac ATTR amyloidosis is a progressive disease as a result of ongoing accumulation of amyloid deposits in the heart, and it's ultimately fatal, usually within about 5 years. Hereditary ATTR amyloidosis is an autosomal dominant condition which is associated with mutations in the TTR gene, of which there are more than 130. It's rare, it's progressive, and also fatal in the absence of therapy. And it's caused by amyloid deposits which accumulate both in the heart, but in the hereditary form of the disease, also in the peripheral and autonomic nerves. So the principle of treatment in all types of amyloidosis is to reduce or attempt to reduce the rate of ongoing amyloid formation. The aim being to achieve a new equilibrium in which the rate of natural amyloid clearance, which is invariably very slow, exceeds the rate of ongoing amyloid formation. The idea being that you then achieve net amyloid regression. Now, reducing the rate of amyloid formation is achieved in all types of amyloid by reducing the concentration of what we call the amyloid fibril precursor protein. In other words, the protein from which the amyloid fibrils are derived. So in cases of ATTR amyloidosis, the subject of today's talk, what we aim to do is reduce the concentration of circulating TTR protein. So in 2018, the RNA interference therapeutics, patisiran and inotersen, often referred to as gene silencers, were approved in the NHS for treatment of hereditary ATTR amyloidosis. And those drugs work by reducing the circulating concentration of TTR, as I mentioned earlier. So here are some anterior whole body SAP scans. So SAP scanning is it's a nuclear medicine technique in which we can image visceral amyloid deposits in various different types of amyloidosis. It's a nuclear medicine technique that was developed in our center. And what we see in patients who are untreated with amyloidosis is we see accumulation of amyloid in the viscera. So, the black bits that you can see on these images get blacker and blacker, and the organs get bigger and bigger as amyloid accumulates. So the two scans here on the left are from a patient with a form of amyloidosis known as AA amyloidosis, in which the protein that makes the amyloid fibrils is a protein called serum amyloid A protein. And if one treats the inflammatory disease, which causes elevation of the SAA protein, if one treats that effectively and thereby suppresses circulating SAA concentration, then what you can see is gradual regression. These scans are taken 2 years apart, and you can see gradual regression of amyloid from the liver and the spleen. 2 years later, less black in the liver and a smaller and less black spleen. And you see exactly the same thing in AL amyloidosis in this middle panel, following suppression of the concentration of the amyloid protein, which in the case of AL amyloidosis is known as a light chain protein. Okay. In ATTR amyloidosis, again, the subject of today's talk, you can see that with this gene silencing drug patisiran, which knocks down TTR concentration in the bloodstream, you can see gradual regression of amyloid from the viscera. So this is amyloid in the spleen in a patient who started patisiran, and a few years later, this is pretty much a normal scan. You can see that the spleen is a lot less black than it used to be. Okay, and I think it's just worth mentioning that, note the time period between each of the scans. Amyloid regression is, very slow at the best of times. And you can see as well from these serial scans that it occurs at different rates in different individuals. Can you still see my, can you see the screen? Yeah. So
Sorry, I'm not sure what's happened there. Let me just see if I can get your screen back.
Shall I share again?
Yeah, can you try to share again?
Maybe I'll try and share again. Are you seeing that now?
Yes, that's perfect.
Yeah, so apologies. Yeah. So this slide demonstrates the importance of the magnitude of knockdown of the relevant protein. Okay, so what I'm showing you here is that in these same 3 types of amyloid that I mentioned earlier, the greater you knock down the relevant protein, the better the outcome. So in patients with AA amyloidosis, this is the relative risk of death in relation to the concentration of circulating SAA protein in the bloodstream. And this is survival in AL amyloidosis in relation to the relative, this is patients at the top here. The blue line is shows patients who've achieved 100% knockdown of the light chain versus 90% knockdown, the gray line, versus between 50 and 90, the yellow line, and no knockdown, the red line at the bottom there. So you can see the direct relationship in these 2 types of amyloid between the magnitude of knockdown of the precursor protein and outcomes. And the same data is emerging in ATTR amyloidosis on the right there. What you see is the change in neuropathy score. These are patients with hereditary amyloidosis and neuropathy. And this is the change in neuropathy score in relation to the knockdown of the TTR protein. And you can see that the patients on, at the bottom of the figure here are the patients with the greatest knockdown of the TTR protein, and they have the greatest improvement in neuropathy score. So direct evidence of a strong relationship between the magnitude of knockdown of the protein and outcomes. And exactly the same is true in cardiac amyloidosis, which is relevant to ATTR amyloidosis. If you knock down the light chain in AL amyloidosis very dramatically, then you can achieve regression of amyloid in the heart. That's been shown using cardiac MRI techniques, which are a way to look at the actual amyloid burden in the heart. So let's turn our attention to the subject of the day. And, essentially, I think what I've shown you is that existing gene silencing therapies knock serum TTR down by about 80%. And they do benefit neuropathy and cardiomyopathy in hereditary ATTR amyloidosis. But some patients on these treatments, unfortunately, continue to experience debilitating symptoms. And, some of them even die from their disease. So what we need is effectively a better treatment. And one of the other problems with these gene silencing therapies is that they require chronic and lifelong administration. So on the basis of the data that I've shown you so far, one would expect that if we're able to knock TTR down more effectively, that will facilitate clinically meaningful symptomatic improvements accompanied by better clinical outcomes. So editing of the TTR gene has the potential to provide a permanent and profound TTR knockdown after a single infusion. So it's a particularly attractive treatment strategy. So in 2012, an American scientist and a French scientist, Jennifer Doudna and Emmanuel Charpentier, showed how CRISPR, which is a naturally occurring biological defense mechanism in bacteria, could be used as a sort of cut and paste tool for editing gene sequences. And they won the Nobel Prize for Chemistry in 2020 for their work. But just to explain in my simple brain, if you like, it really is like a cut and paste tool. That is the idea, that you can cut and paste gene sequences in the human genome. So NTLA-2001 is an investigational CRISPR-Cas9 based in vivo gene editing therapy. And it consists of a single guide RNA, and that is the molecule that targets the relevant gene. In this case, it's a single guide RNA that targets the human TTR gene. And it also consists of a human optimized mRNA sequence of strep pyogenes Cas9 protein encapsulated in this lipid nanoparticle. So if you like, the guide RNA provides the target. It means that the drug goes to exactly the precise region in the genome or in the gene that one wants it to target. And the Cas9 protein, in effect, are like the scissors. And what's important and relevant, of course, for hemophilia is that this therapeutic platform is modular. It is actually extremely easy to produce a guide RNA molecule that targets any gene in the body. So this slide summarizes the mechanism of action of NTLA-2001, which is administered as a single intravenous infusion of nanoparticles. And as you saw what the nanoparticles consist of. After entering the circulation, the lipid nanoparticles are taken up by the LDL receptor on the surface of hepatocytes. And after endosomal release, the Cas9 mRNA is translated, producing the Cas9 enzyme, as I said, the scissors, if you like, and the enzyme interacts with the single guide RNA to form what is known as a CRISPR-Cas9 complex. That complex enters the nucleus, and a 20 nucleotide sequence at the 5' end of the guide RNA selectively binds to its complementary sequence on the patient's DNA, leading to what's called a double-strand break in the targeted TTR gene sequence. In other words, you get cleavage, you get the scissors in both strands of the targeted TTR gene sequence at a very precise, in a very precise place, if you like. And then something, endogenous DNA repair occurs through a process called non-homologous end joining, which leads to what we call indels, insertions or deletions of nucleotides in the TTR gene, which prevents production of the TTR protein. Okay, so that's the mechanism of action of NTLA-2001. So one of the major concerns surrounding gene editing therapy is the potential for what is known as off-target editing. In other words, one wants to edit very specifically the gene that one wants to edit, but what you don't want to do is start editing or altering genes which you didn't intend to edit, and that's known as off-target editing. So the scientific team from Intellia, the company that developed NTLA-2001, undertook a rigorous approach to select a single guide RNA to ensure both potent on-target, in other words, potent editing of the gene that one wants to target, in this case, the TTR gene, and undetectable off-target editing. And that's a very important, iterative concept to improve this technology, which has been crucial to it actually being administered to humans. So first, they used computational analysis to identify sites in the TTR gene that are susceptible to CRISPR-Cas9 mediated knockout. Then the team eliminated sites in the gene that contain disease-causing TTR gene mutations or prevalent single nucleotide polymorphisms. The idea being that a single drug could knock down or knock out the TTR gene in a patient regardless of what mutation they carry or whether or not they carry a mutation. Okay. Next, they synthesized a pool of guide RNAs, and they tested each of those guide RNAs for editing efficiency and potential off-target editing using a variety of methods, including in human cells and in animal models. And finally, they validated and selected the guide RNA with the highest on-target editing efficiency and the lowest off-target editing potential. So this figure illustrates the effect of NTLA-2001 on primary human hepatocytes. So importantly, no evidence of off-target editing was detected even at NTLA-2001 concentrations threefold greater than the EC90, which is shown by this vertical line here. That's the concentration at which one achieves 90% reduction in TTR production. This is editing of the TTR gene, and you can see no off-target editing. So these data really demonstrate the high specificity of NTLA-2001 for the targeted site in the TTR gene. So the preclinical in vivo studies were conducted in cynomolgus monkeys. You're using a surrogate lipid nanoparticle incorporating a single guide RNA molecule, which is specific to the monkey TTR gene. And what this graph shows is the mean percentage reduction from baseline in serum TTR protein concentration in monkeys receiving the LNP at doses of 0, 1.5, 3, and 6 milligram per kilogram of the RNA cargo on day 0 and followed up for a total of 367 days. Note, a single dose at 3 milligram per kilogram achieved a durable reduction in circulating TTR protein of approximately 95% after day 28. Safety studies established a dose range at which no adverse effects of NTLA-2001 were observed, with near complete sustained reduction in serum TTR following a single administration. At higher doses, the adverse effects in the monkeys were dose dependent, but they were transient, reversible, and monitorable. So NTLA-2001 is being studied as a two-part open-label multi-center study in patients with hereditary ATTR amyloid polyneuropathy or ATTR amyloid cardiomyopathy. And the primary objectives of this first-in-human study are obviously to evaluate safety, tolerability, pharmacokinetics, and pharmacodynamics, the latter by measurement of circulating TTR concentration. So I'll present the part 1 data from both the polyneuropathy and the cardiomyopathy arms of the trial. So first, polyneuropathy. Part 1 was a single ascending dose design with a minimum of 3 patients in each of up to 4 dose cohorts. Part 2 is a single dose expansion study. So I'll report the first 15 patients with hereditary ATTR amyloidosis and polyneuropathy from the dose escalation cohort here in part 1. So these are the demographics and baseline disease characteristics of those first 15 patients. I won't bore you with the details, which are not terribly relevant, other than to say that these patients typically had fairly early stage disease. So they didn't have terrible neuropathy or terrible cardiomyopathy consistent with a phase 1 study. But the other important point is that a range of different mutations were represented amongst patients, those first 15 patients. And here is the early safety data. So NTLA-2001 was generally well tolerated in the acute phase, with the majority of adverse events being grade 1, i.e. mild in severity and mostly unrelated to treatment. There were a number of infusion-related reactions, but they were all considered mild, didn't require dose interruption, and they all resolved without clinical sequelae. and all of the patients received a complete study dose of NTLA-2001. There was a single serious adverse event, which was that of intractable vomiting in a patient who was actually known to have very significant pre-existing amyloid-related gastroparesis from autonomic neuropathy. This patient received the 1.0 milligram per kilogram dose of NTLA-2001, but his symptoms settled very quickly, and the patient has since been extremely well. This just summarizes the adverse events in more detail. As I say, the most common adverse events were mild and transient headache and mild infusion-related reactions, usually manifesting as fever or chills, but they were short-lived and recovered. And this is the surrogate efficacy data, if you like, the exciting data from a disease treatment perspective. And what you see is that at day 28, a dose-dependent reduction in serum TTR concentration with a reduction of in the high 80s or 90s at these higher doses of the drug. So very marked knockdown of TTR, circulating TTR concentration in the bloodstream. And here you see that at the higher therapeutically relevant doses, the deep serum TTR reductions were sustained through to between 6 and 12 months, the last visits that I'm free to report, if you like. So patients in the cardiomyopathy arm either have hereditary ATTR amyloidosis or wild-type ATTR amyloidosis, obviously with cardiomyopathy causing heart failure, and they have NYHA class 1 to 3 heart failure. Again, part 1, there was a minimum of 3 patients in each of 3 cohorts. An NYHA class 1-2 cohort administered 0.7 milligram per kilogram. An NYHA class 3 cohort administered 0.7 milligram per kilogram. And an NYHA class 1-2 administered 1.0 milligram per kilogram. Based on safety and the PD profile at 0.7 milligram per kilogram, further dose escalation to 1.0 milligram per kilogram in NYHA class 3, in other words, very advanced heart failure patients, was not undertaken. Part 2 is a dose expansion cohort in a similar vein to the polyneuropathy arm. And I'll report data on all 12 patients enrolled in the cardiomyopathy arm. I should say that the cardiomyopathy arm started after the polyneuropathy arm, which is why the 0.1 and 0.3 milligram per kilogram doses that are, that I showed in the polyneuropathy arm were not included in this cardiomyopathy arm of the trial. So it was really just in, if you like, therapeutically relevant doses. So this table summarizes the demographics, and really the important point is that the 50% of the patients had NYHA class 3, so very advanced heart failure, and the median NT-proBNP, a biomarker of heart failure, if you like, in patients with amyloid in their hearts, was markedly elevated at 2,400. The normal range is less than 300 or thereabouts. So I will summarize the safety data again. So again, in patients with predominant cardiomyopathy, NTLA-2001 was generally well tolerated across all dose cohorts through the follow-up period. The majority of adverse events, again, were mild in severity. The most commonly observed adverse event, again, was infusion-related reaction in 2 patients. 3 of the 12 patients reported no AEs, and 8 reported only mild or moderate AEs. All patients, again, completed their infusions and all remain on study. A single grade 3 infusion-related reaction was reported at the 0.7 milligram per kilogram dose in a patient with NYHA class 3 heart failure, but that resolved without any clinical sequelae. The 0.7 milligram per kilogram, the NYHA class 3 cohort were expanded to 6 patients, as per protocol, if you like, and no additional patients in that cohort reported a treatment-related adverse event higher than grade 1. And lastly, there were no clinically significant abnormal laboratory findings. And again, this table summarizes the most common adverse events that occurred in at least 2 patients. And again, they were predominantly transient, reversible, and mostly grade 1. And what you see here is the slide summarizing the pharmacodynamic effects and the durability from baseline to the latest measurement. The patients in the MYHA class 1, 2.7 milligram per kilogram cohort have now reached 6 months, and they've achieved a consistent, sustained response with a mean 93% reduction in circulating TTR in the MYHA class 3.7 milligram per kilogram and the MYHA 1/2 1.0 milligram per kilogram cohorts. The mean reductions were 94% and 92% respectively, and they were observed at 4 months of follow-up. Importantly, by day 28, all 12 patients had achieved at least 90% TTR knockdown. So the conclusions from this interim study data are that deep, consistent, and durable TTR reductions were achieved at both 0.7 milligram per kilogram and 1.0 milligram per kilogram doses. The mean TTR reductions were greater than 90% across both doses by day 28 and sustained to last follow-up. NTLA-2001 was generally well tolerated. The majority of adverse events, as I mentioned, were mild, and there were no clinically significant laboratory findings of concern, as it were. The results were similar across both dose levels and across both NYHA heart failure class cohorts, and the data presented in the cardiomyopathy cohort are consistent with the data in the polyneuropathy arm of the trial, which, as I mentioned, was conducted earlier, if you like, and they basically further support the earlier findings and the promise of CRISPR-based in vivo gene editing in humans. More specifically from an amyloidosis perspective, the deep TTR reductions and the sustained TTR reduction observed in this study has the potential to bring about not only clinical stability, but genuine clinical improvement in patients with ATTR amyloidosis, which is hitherto, as I mentioned earlier, been a universally progressive and fatal condition. So in considering the potential future clinical applications of CRISPR-based gene editing therapy, perhaps the first thing to think about is why was ATTR amyloidosis chosen, if you like, as the prototypic disease? Well, firstly, there's an unequivocal link between the protein and the disease, which is also true for hemophilia. Secondly, the circulating TTR protein is exclusively produced in the liver. And targeting the liver using a lipid nanoparticle is certainly something that, as I've shown you here today, is eminently achievable, if you like. Thirdly, knockdown of the TTR protein was already known to be safe, and that was based on the fact that it's known that there is significant overlap in the physiological functions of TTR, and also our extensive experience with the gene silencing drugs, which, as I mentioned to you earlier, knock down TTR by about 80%, if you like. And they haven't been associated with any concerning safety signals. So we knew that knocking down TTR was a safe thing to do. And we also knew that knocking down TTR was clinically beneficial. We know from the trials and our experience with the gene silencing drugs that knocking down TTR is associated with clinical benefit. And lastly, and perhaps very relevantly to hemophilia, is that the knockout editing technology in terms of gene editing is relatively straightforward. And what I mean by that is that what we are trying to do here is just to knock out the gene. In other words, induce those double strand breaks that I mentioned to you earlier, and therefore, stop production of the TTR protein from liver cells. So it's a relatively speaking, all one needs to do is knock out the gene, if you like. So let's think about future potential CRISPR applications and technologies. Well, I've told you knockout in vivo gene editing is possible. I've shown you that today, I hope. And it's already being trialed in several other diseases mediated by genes that are expressed usually or solely in the liver. An example is hereditary angioedema, but there are several other examples in which drugs are actually being administered in vivo. Editing is being administered to humans in various trials. So that, if you like, is now a technique which one might argue is to some extent conquered, if you like. I think it's early to quite state it like that, but certainly delivery of a gene editing drug to the liver in vivo is possible, as I've shown you. For hemophilia, one needs knock-in editing strategies. In other words, one needs to knock in the either factor 8 or factor 9 gene. As you will know, many of you will know far better than I do previously, the idea was to transduce the factor 8 or factor 9 gene into hepatocytes using viral vectors. But the AAV vectors have limited packaging capacity. That's particularly relevant to the hemophilia A gene, which, as you know, is a large gene. And transgene expression, particularly in hemophilia A, appears to decline such that treatment may not always be necessarily lifelong. In addition, liver-targeted delivery of AAV may be associated, as you will all be aware, with transaminitis. So another possibility is to use CRISPR-Cas9 to mediate targeted integration of the factor 8 or factor 9 cDNA into the intron of a host cell gene, such as the albumin gene, or alternatively, to achieve targeted repair of the mutant gene. And the advantage of these approaches is the potential for lifelong benefit due to, if you like, endogenous gene expression. There has been success with this approach in mice, for example, in a mouse model of hemophilia A, in which exon 1 of the factor 8 gene was deleted. Donor DNA containing a homologous exon 1 sequence and CRISPR-Cas9 were packaged together into a viral vector and used at relatively low dose, injected intravenously, resulting in integration into some hepatocytes of the correct exon 1 sequence via a technique known or via a process known as homology-directed repair, which, if you like, is an accurate way of joining the ends that are created by the scissors, by the CRISPR, the double strand breaks that I mentioned. As I mentioned earlier, for knockout gene editing technology, we rely on a process known as non-homologous end joining. And that's a rather inaccurate process, which means that you get these indels, which I mentioned earlier. In other words, insertions and deletions of nucleotides, which means that the gene, the protein is no longer encoded for by the gene, if you like, because the gene has not got the perfect sequence. But homology-directed repair is a far more accurate way in which one could potentially use a homologous a section of gene to direct repair of a particular gene sequence so that that gene sequence achieves, if you like, wild type sequence. So that's the idea. The technology, is not there yet, but that is the idea, and that is, I think offers certainly hope for the future. There are various other future applications which include base editing, single base edits, for example, in sickle cell disease. There's prime editing, which is editing of either single bases or longer stretches of DNA. There's something called multiplex editing, which is particularly interesting potentially interesting for cancer, because one can introduce multiple single guide RNAs, if you like, package them within one drug, and those different guide RNAs may target different genes. And as you know, multiple genes may be involved in cancer risk, etc. And then there is also CRISPR activation, and there's CRISPR-A and CRISPR inhibition, CRISPR-I, to turn genes on and off. So there are various technologies, none of which have yet been applied in humans, but there are a whole range of techniques about which I will be, as I mentioned earlier, I am by no means an expert, but at the same time, there are various technologies which offer great hope for CRISPR-Cas9 gene editing in vivo for the future. So I would just like to finish by summarizing. In vivo knockout editing looks extremely promising, and I hope you'll agree with me on the basis of the trial that I've shown you. The safety data is certainly extremely encouraging, and there have been really no concerning safety signals thus far. And obviously the permanence of the TTR knockdown and the permanence of the effect is equally encouraging. I should say that during the preclinical studies, which I didn't actually mention to you earlier, a guide RNA was directed against the mouse TTR gene and given to mice, following which 2/3 of their livers were chopped out, if you like, and following regeneration of the liver, the team were able to show that TTR levels remain suppressed. So that tells you that the gene editing, if you like, is targeting liver progenitor cells, and explains, or certainly goes some way to explaining, the permanence of the effect that we are observing in the patients and that was observed in the non-human primate study. And as I've mentioned to you, knock-in technology is required for hemophilia. I think there are obviously exciting possibilities for permanent gene correction and or integration within the genome. CRISPR-Cas9 based therapies may need to be mutation specific, but there is considerable ongoing work required, I think it's fair to say, to maximize integration efficiency, accurate integration in terms of the position within the genome, and obviously to minimize the risk of off-target editing, as I touched on. So I'd like to just finish by thanking firstly Intellia and Regeneron, that is the company that designed NTLA-2001, which is the drug that I've been talking to you about, and also thanking the patients, their families, and my co-investigators, and also all my colleagues at the National Amyloidosis Center, many of whom have been involved with this trial and many other trials that we've been involved with in amyloidosis. So thank you very much for listening.
Thank you very much, Professor Gilmore. I think we just are going to try and add a few panelists to speak with you. I think we've got Len Valentino there.
Yep, I'm here. Thank you, Dr. Gilmore. This was a great presentation. Really appreciate you doing this. Maybe while Fiona is working on that, I can ask you a question about age limitations. So is there, currently AAV is limited at the lower end by regulatory considerations, but potentially at the upper end of the age groups by potentials for toxicity, et cetera. So do you see that this technology will have similar age limitations?
I suspect not, actually, and you can see that. You can see that in the ATTR amyloid cardiomyopathy is actually a disease of older age, and although the hereditary form frequently involves young individuals, the wild type, the non-hereditary version, which predominantly affects the heart, is typically a disease of older age. And in terms of gene editing, there's no reason really to to limit the age, the age cutoff to younger patients necessarily. In fact, one might even argue, and this is purely speculative, but one might even argue that if there was going to be a risk of off-target editing, it might be better to treat someone rather later in life than necessarily treating someone when they're very young and might develop some horrible effect of off-target editing that was unexpected. Certainly I would say that potentially for the early stages of this new technology, which is where we are at the moment.
Yeah, so maybe to follow up with another question, and then I'm going to introduce Mr. Mark Skinner. But, you mentioned off-target effects. So what is being done both in the laboratory as well as in the clinical trials to monitor for off-target effects? And are we able to really determine with any sense of precision whether there are off-target editing effects?
Yeah, it's a very important question. And obviously in the non-human primate study that I described to you, there was a very extensive search with biopsies of various organs for, off-target effects, particularly extensive search, obviously, in liver tissue, which is where the vast majority of this drug goes. So, there was very extensive search for off-target editing in, on the basis of the non-human primate study. In the human study, it's far more difficult because we didn't feel that we could justify liver biopsies in patients having this treatment. And so the monitoring for off-target effects is really clinical monitoring, to be honest. And, so far, touch wood, we have no indication of off-target editing or off-target effects. Which is consistent with the preclinical data that I showed you, which suggests that the drug is highly specific for the TTR gene within both, within hepatocytes in vitro and also, as I say, in the non-human primate study. But at the same time, it's far from perfect. You talked about precision, and as you can tell, this is not a particularly precise way of looking for potential off-target editing.
Yeah, thank you. So we've also got a panelist, Mr. Mark Skinner. I'll let Mark introduce himself to you so you know who's asking you a few questions. But we're trying to get one other panelist in here as well. So go ahead, Mark.
Hello, Dr. Gilmore. Thank you for a really interesting presentation and thinking about the future and that day when there could be a, quote, cure for hemophilia is really exciting. I am an individual that lives with severe hemophilia A, so I followed this space for quite some time, do a lot of research, sort of both focusing on burden of disease and outcomes. I'd like to follow up just a little bit on Dr. Valentino's question about the age limitations. The current gene therapies, although bringing the potential for significant advance, have other limitations. In the hemophilia A space, prior exposure to the AAV or other limitations may come into play. And so what I think the community at large may not realize is the gene therapy is not available for everyone. Do you see those same kinds of limitations? Will this would this potentially be limited to somebody with a specific mutation or an inversion or something else or could it have broader applicability without as much need for, pre-screening, which I guess could then theoretically lead to greater predictability in terms of those who might ultimately benefit?
Yeah, I mean, I suppose to answer your question, in amyloidosis, I would imagine not, because what we're trying to achieve in amyloidosis is, in one sense, very simple compared to, what is necessary to successfully treat hemophilia. And as I mentioned, all we really need to do, if you like, is induce, a double-strand break in the right place, hopefully without inducing a double-strand break in another gene. So in effect, all we want is to direct the scissors, the Cas9 protein, to the right place in the genome and be able to deliver it to the right cells. Delivering it to the right cells in the liver, the hepatocytes, which is where TTR is produced, has really been achieved. We've shown that. And, chopping the TTR gene very specifically seems to have been achieved. Well, it has been achieved. I suppose, as alluded to earlier, we can't completely say at the moment it's completely specific. We don't know that yet, but certainly every indication is that it is specific. I think, to answer your question, I think for, in hemophilia, and I'm certainly no expert in hemophilia, it's a very different challenge, isn't it? Trying to insert, there you're trying to, allow the correct piece of DNA, a homologous piece of DNA, to be inserted in the correct place to essentially either correct a gene defect or to express either factor VIII or factor IX, and to be incorporated into the host genome. So that's a very, very different challenge. Having said that, should it be limited to, patients of a certain age rather? Not necessarily. I wouldn't think that age in itself should be a reason to necessarily limit this form of gene therapy, if you like, or gene editing, other than obviously on the basis of cost, which is, which does tend to be, a consideration for, for regulatory authorities or certainly approving authorities in various countries.
Can I ask just a follow-up? So that was sort of the who might be eligible in terms of limitations. Thinking about sort of post-gene editing, we also are aware there is a significant burden, particularly at year 1, for the follow-up and monitoring. And one of the things that we seek to avoid with AAV is germline transmission. And so while there's ethical issues around gene editing, which we'll leave for another day, but what's the post-gene editing burden like? Is the same degree of intensity there? Or what would it mean for individuals of sort of childbearing age, whether they're planning to have children or otherwise?
Yeah, so I should just say that in the preclinical studies, there was no evidence of germline editing, and that's an important point that you've touched on. And as you alluded to, the regulatory authorities will not allow you to deliver a drug that induces germline gene editing. So that's an important point. The monitoring following gene editing in our disease, as I say, the knockout gene editing is actually pretty minimal. There was obviously considerable monitoring at the start. We had the first patient ever to receive any in vivo CRISPR-Cas9 based gene editing therapy in our unit, and you can imagine that there was obviously a degree of nervousness around that infusion. So obviously the first few patients were monitored extremely carefully as inpatients following their single infusion, and they continue to be monitored carefully, obviously, within the first 24 hours. But, we are much more relaxed now after having treated about 50 patients and not had any, problems of significance, if you like. following the actual infusion and the patient's discharge 24 hours later, if you like, then the monitoring now is very minimal. It's sort of 3 monthly monitoring for any adverse events, as well as monitoring of their amyloidosis, if you like.
Thank you.
Yeah, maybe I'll ask another question about the applicability of this technology and gene editing in general to women who may have, whether it's amyloidosis or hemophilia or other disorders. Do you see that that's going to be a possibility into the future?
Yes, I do. I mean, obviously, it's easier, isn't it, to propose it in a disease that affects women of after childbearing age. So I think that's certainly easier to propose. But at the same time, I think that if one really sees no evidence with the experience that we will gain with various gene editing therapies, in the near future, at least knockout gene editing therapies, if one sees really no evidence of editing, in the germline, then one could certainly be very reassured by that. It wouldn't mean to say that you wouldn't counsel patients, and it wouldn't be an extensive consent form to sign. So I wouldn't pretend to ignore the risks that the putative risks that we're talking about, but at the same time, I think the data as we have it at the moment, and hopefully ensuing data will be encouraging enough that we will be comfortable delivering it to women of childbearing age at some time in the future.
Yeah, another population that we're particularly interested in is people who live in low middle income countries. So hemophilia is a disease that is benefited from technology, and the current standard of care in the developed world is excellent. However, the standard of care outside of the developed world is particularly low. So, bringing gene editing or any form of novel intervention to the developing world is always something that we're keenly interested in, where the risk benefit may be very different, in that discussion. So what are your thoughts on this technology in the developing world?
Absolutely. And that's a subject that's very close to my heart, because we have the same problem in amyloidosis. We have high cost drugs. The gene silencing therapies that have been approved are approved in certain countries and not others. And it's the issue of cost is certainly a huge one across all therapies for for ATTR amyloidosis, as they undoubtedly will be for hemophilia. So I think those are definitely concerns. Access to these drugs is going to prove challenging. I think in terms of the actual safety, if you like, I would hope that the safety, which unfortunately I think you're right, the safety studies will be conducted in probably mostly in developed countries, and I hope that the safety profile of various gene editing therapies will be sufficiently confident about it, that in terms of, for example, a less developed healthcare system where monitoring may not be equally easy, if you like, wouldn't necessarily put one off delivering this treatment.
Yeah. Dr. Gilmore, thank you so much for introducing gene editing to the hemophilia and the bleeding disorders community. Even though the disease that you're addressing is not necessarily relevant, the technology is extremely exciting and obviously very applicable to our community. So thank you very much.
You're very welcome. Thank you very much for inviting me.
Thank you. Thank you very much, Professor Gilmore, and thank you, Dr. Skinner and Dr. Valentino, for your questions as well. I'd like to thank you all for joining us this afternoon. It was really a very exciting talk, and I think we've all learned a lot. Just note that this recorded webinar will be available on Friday, July 21st at hemophilia.org under the events tab with all of our archived webinars. Also available in the events tab is our upcoming schedule for our weekly Wednesday webinar series. Next week, we will hear from the World Federation of Hemophilia, and they will be offering us an early view of the new shared decision-making tool that they will soon be launching. So thank you very much for joining us this afternoon, and have a wonderful week.